Assalamu alaikum. Today we are going to study some main topics of subject biology, class first year. These video lectures are also useful for MBBS test. These lectures are in very simple words by which anyone can take help. If I miss anything in this video or you don't understand anything, so comment in the comment section. Let's move to our lecture. Let's explore some questions of biology with answers. How is the cell wall of plant studied? The cell wall of a plant cell can be studied using techniques such as microscopy, immunofluorescence, and biochemical analysis. These methods allow researchers to visualize and characterize the composition, structure, and function of the cell wall. What techniques are used to study plasma membrane? Techniques commonly used to study the plasma membrane include electron microscopy, fluorescence microscopy, and biochemical assays. These methods enable the investigation of membrane proteins, lipid composition, and membrane dynamics. How is the cytoplasm of cell analyzed? The cytoplasm can be analyzed using various techniques including self-fractionation, microscopy, and biochemical assays. Self-fractionation allows the separation of cellular components, while microscopy and biochemical assays enable the study of cytoplasmic structures, molecules, and metabolic processes. What methods are used to study organelles? Organelles can be studied using techniques such as electron microscopy, immunofluorescence, and molecular biology approaches. Electron microscopy provides detailed images of organelle structure, while immunofluorescence allows for the visualization of specific proteins within organelles. Molecular biology techniques such as gene knockdown or overexpression can help elucidate organelle function. How are prokaryotic cells analyzed in cell biology research? Prokaryotic cells are analyzed using techniques like microscopy, molecular biology, and biochemical assays. Microscopy allows for the visualization of prokaryotic cell structure, while molecular biology techniques help study gene expression, regulation, and protein function. Biochemical assays assist in characterizing metabolic pathways and enzymatic activities in prokaryotes. What techniques are used to study eukaryotic cells? Eukaryotic cells are studied using a wide range of techniques including microscopy, light microscopy or electron microscopy, molecular biology techniques, example PCR or DNA sequencing, cell culture, and genetic manipulation. These approaches allow researchers to investigate various aspects of eukaryotic cell biology, such as structure, function, and molecular mechanisms. How are the components of cell wall identified? The components of cell wall can be identified using techniques such as biochemical analysis, mass spectrometry, and genetic approaches. Biochemical analysis involves the extraction and characterization of cell wall components, while mass spectrometry allows for the identification and quantification of specific molecules. Genetic approaches involve studying mutants or genetically modified organisms to understand the role of specific genes in cell wall formation. How can the fluidity of plasma membrane be measured? The fluidity of the plasma membrane can be measured using techniques like fluorescence recovery after photobleaching and fluorescence anisotropy. FREP involves bleaching a small area of fluorescently labeled membrane and observing the recovery of fluorescence over time, which indicates membrane fluidity. Fluorescence anisotropy measures the rotational mobility of fluorescently labeled molecules within the membrane, providing information about its fluidity. How is the localization of organelles determined within the cell? The localization of organelles within cell can be determined using techniques such as fluorescence microscopy, immunofluorescence, and live cell imaging. Fluorescence microscopy allows for the visualization of organelles using fluorescently labeled markers or antibodies. Immunofluorescence specifically targets and visualizes proteins within organelles. Live cell imaging enables the dynamic observation of organelle movement and interactions in real time. What techniques are used to differentiate between prokaryotic and eukaryotic cells? Techniques used to differentiate between prokaryotic and eukaryotic cells include microscopy, genetic analysis, and biochemical assays. Microscopy allows for the observation of cell size, complexity, and the presence of membrane-bound organelles. Genetic analysis can reveal the presence of distinct genetic features in prokaryotes, example circular chromosomes, plasmids, and eukaryotes, examples linear chromosomes and introns. Biochemical assays can detect unique cellular components such as peptidoglycan in prokaryotes and a variety of membrane-bound structures and organelles in eukaryotes. What is micrometry? Micrometry, also known as micrometer measurement, refers to the process of measuring small distances, dimensions, or features using a device called a micrometer. A micrometer, also called a screw gauge, is a precision instrument used to measure the thickness, diameter, or length of an object with high accuracy. What is endoplasmic reticulum? The endoplasmic reticulum is an organelle found in eukaryotic cells, which are cells that have a nucleus and membrane-bound organelles. It is a complex network of membrane-enclosed tubules and flattened sacs called cisternae that extends throughout the cytoplasm of the cell. Types of endoplasmic reticulum. There are two types of endoplasmic reticulum, rough endoplasmic reticulum and smooth endoplasmic reticulum. The rough endoplasmic reticulum is so named because its surface is studded with ribosomes, giving it a rough appearance when viewed under a microscope. The ribosomes on the rough endoplasmic reticulum synthesize proteins that are destined for secretion, incorporation into a cell membrane, or for use within other organelles. The rough endoplasmic reticulum is involved in protein folding, modification, and quality control processes. It plays a crucial role in protein synthesis and trafficking within the cell. Second type of endoplasmic reticulum, smooth endoplasmic reticulum. The smooth endoplasmic reticulum lacks ribosomes on its surface and appears smooth under a microscope. It has various functions including lipid metabolism and synthesis, detoxification of drugs and toxins, and storage and release of calcium ions. The smooth endoplasmic reticulum is involved in the synthesis of lipids such as phospholipids and steroids, and it participates in the detoxification of harmful substances in the cell. Function of endoplasmic reticulum. The endoplasmic reticulum has various important functions within the cell. Protein synthesis and folding. The rough endoplasmic reticulum is involved in the synthesis of proteins. Ribosomes attached to rough endoplasmic reticulum translate mRNA into protein chains, which are then translocated into the endoplasmic reticulum lumen. The endoplasmic reticulum provides an environment for proper folding and modification of proteins before they are transported to their final destinations such as cell membrane or other organelles. Protein modification. The endoplasmic reticulum plays a crucial role in post-translational modifications of proteins. This includes processes like glycosylation, where sugar molecules are added to proteins, as well as disulfide bond formation, which contributes to protein stability.
These modifications help ensure proper protein structure and function.
If you liked this video, then please like, subscribe, comment and share our channel Dr. Raza Academy and Educational Consultancy.